going to spend some time this morning taking you through the work that we've done for the past few years. You heard a talk earlier this morning by Sally Dickinson that really described the clinical implications of some of what we've begun to do. And I'm not, so bear with me. I'll disclose first that we did start a company based on some of this technology, and so although entrepreneurship is a good thing, for those of you who are students, take what I say with a grain of salt, acknowledging that I have a conflict of interest there. So I want to talk about cell-based studies and where we really are in the field, beginning with the concept that today there are really a number of studies underway throughout the progression of ischemic heart disease, beginning with coronary atherosclerosis, proceeding through acute myocardial infarction, through post-myocardial infarction heart failure to end-stage disease. And that as of today, there are about 442 studies currently registered at clinicaltrials.gov under stem cells and heart, and about 198, almost 200 of those studies are actively recruiting. And you can see that there are a number of different cell types involved, from bone marrow mononuclear cells to mesenchymal cells to endothelial cells to skeletal myoblasts, and more recently cardiac-derived progenitors and fat-derived cells. And in fact, the first cell type that's probably going to be approved or receive a CE mark is the fat-derived cell here in Europe. If you think about a few years ago, 2009, there were only 95 clinical studies underway. So today, that has increased about fourfold, and you can see that the types of patients have progressed as well, from acute coronary syndrome, large number, to heart failure, to chronic heart disease. And the cell types that are most often studied are bone marrow mononuclear cells, with the diploblasts being the least well studied or studied in the lowest percentage, but as I said, those are probably the ones that are going to actually be approved first. And you can see that we've moved from early studies on just stem cells to even the use of stem cells with other existing novel therapies like cardiac resynchronization therapy, and more recently the use of human cardiac-derived stem cells to treat ischemic cardiomyopathy. And I'll just point out again that although there are a number of studies, these are not overwhelmingly positive. They're moderately positive studies, and I think the future is going to be really understanding how to manipulate the process of endogenous repair with our own cells, using other cells, growth factors, microRNAs, etc. And in addition, we need new cell-based options. And at the American Heart Association meeting this past November, 2 new clinical trials were described, the Scipio trial and the Caduceus trial, with cardiac-derived cells, and I'm happy to talk about those later if anyone has any questions. But we're of the opinion that cell therapy, while beneficial, may not necessarily ever suffice for end-stage heart failure, and that's because I don't think it's ever going to be possible to take cells and transplant them into a thinned-out, scarred region of myocardium and fully recapitulate vasculature and functioning muscle, much as we may try. And so we said, let's take a different approach. And you see a postcard that used to be on my office wall, and I actually gave it to someone recently who's doing more of this than I am, and many of you probably know who she is. Her name is Katie Piper, and she's from the UK, came to visit my lab, and is really an advocate for novel therapies going forward. But I believe in trusting our crazy ideas and trying to build a new solution, and so we said, let's naively see if we can build a new heart. And we started this a few years ago, and because I'm not a tissue engineer, I said, what do we need to build an organ? Well, we need cells, but we need cells in a large quantity, and stem and progenitor cells are one opportunity, and they exist in every organ or tissue, but they don't exist at hundreds of billions, which is what I would argue we're going to need to do this going forward. But we don't just need cells. Beating cells in a dish don't constitute a heart. We need a scaffold, and not just an inert scaffold, but a scaffold that supports organ function. And then if we're going to build anything complex, we need a vasculature that can feed the organ or tissue so that we can build, if we're going to build a heart, we don't want to build an infarcted heart, we want to build a heart that can be perfused. So we need vasculature to feed the cells. And then we really need physiology. And I'm going to come back to the fact that I think physiology is critical to mature any cells that we put on a scaffold. So what are the cell options? You've all heard this. We know that there are adult tissue-derived options, and I would go so far as to say that going forward for any complex organs, we're probably going to use bone marrow or blood-derived cells to regenerate the vascular components, and then other probably mesenchymal cell types from skeletal muscle, fat, or heart to begin to generate the cardiac parenchymal cells that we need. Obviously, adult iPS cells are another option going forward, and allogeneic cells as well as embryonic cells. Now, we've focused primarily on an adult heart type cell that you can see on the right can actually give rise to beating cells in a dish. And we can isolate very small numbers of these. The pointer doesn't work that well, but we can start with one, grow them to large numbers, and we can get these to give rise to smooth muscle cells, endothelial cells, and these beating cells in a dish. But as I said, cells alone don't make an organ, so we need a scaffold. And you've already heard a very elegant approach, and an approach that we took a couple of years ago was the concept that nature had already created this perfect scaffold. This is a pig heart, and you can see obviously multiple chambers, the vasculature, you see the coronary arteries here, the great vessels, and we said perhaps we can take advantage of this scaffold and build one that we can use. So we developed a perfusion-based process whereby we could remove all of the cells from a complex organ or tissue. And we did that by actually using the vasculature to infuse detergents over time. And we published this a few years ago, and removal of the cells with detergent essentially gets rid of all cells and DNA, but preserves the extracellular matrix components. And I want to just point out that we retain a lot of biological signals, including glycosaminoglycans and some growth factors. And because we use the vasculature to deliver these cells, we have open vessel lumens at the end of the period and relevant mechanical properties. And in addition, this is a decellularized pig heart. You can see we retain exquisite microstructure. You see the chordae here, the valve. You can see the whole heart here. And if you look at the left ventricle, it essentially looks as if we took a tube of toothpaste and squeezed it and essentially extruded all of the cells. So we believe that we have an architecture then that's functionally relevant into which we can begin to put cells again. Now I want to stop for a minute and make a point that this matrix does retain a lot of the important properties, and in fact, when we look at the mechanical properties of the matrix, here you can see left ventricle and right ventricle, and we look at, in this case, ultimate tensile strength, but the same thing is true for distensibility or stretch. And you can see that there's a difference in two different directions, a circumferential and longitudinal direction here. In addition, there's a difference by region. So the myocardium, even absent the cells, the matrix retains the mechanical properties of anisotropy so that we can get appropriate squeeze when we put cells back in. And this matrix has a differential impact on cells. If we put the same cells on left ventricular matrix or right ventricular matrix and measure force, you can see that we get a difference in force of the same cells depending on the region where we transplant the cells. And I'm not going to talk about it today, but I'll also mention that we see differences based on whether or not we derive the matrix from male or female animals, and whether or not we use male or female cells on the matrix. And so I only say that because all of you who are doing pharmacologic studies and other things should realize that there are sex differences that begin to impact what we're doing. Now, if we want to build a complex tissue, we obviously need vasculature, as I said. And you can see here a Masson's trichrome image of our decellularized heart showing open veins and arteries here from a rat heart, and a bisected rat heart that's translucent so that you can see the retention of the vasculature. If you look at the ultrastructure, you can see, much like the cadaveric heart, we have an open coronary artery here at the ostium, and the only difference is the absence of these fuzzy cells that actually line the vessel. So we wanted to show that more than just having open vessels, that these were patent, so we did a heterotopic transplant of these, and you can see here that when we do the heterotopic transplant and release the aorta, that in fact the vessels are patent and that we can perfuse this construct. So that gave us confidence that we could begin to go forward. However, not surprisingly, when you think about it, this is naked matrix, and much like any naked matrix, coronary artery or anything else notwithstanding, after a week, what we found is that this matrix was thrombogenic. It clotted. So the question was, could we actually impact this? Now, what we found was going on as we looked at this clotted matrix was when we harvested the heart that we had transplanted, there was essentially a competition between re-endothelialization. So this construct recruited endothelial cells into the vasculature, but it was competing with clotting that was going on over time, and so we couldn't actually keep up. So we said, can we actually add back endothelium? And you see here a rat heart where we've infused di-I and di-O labeled rat aortic endothelial cells into this and let them grow for 7 days. And what you can see is that we have cells that actually reline both small and large diameter vessels. You can see here they're PCNA positive. They're also eNOS positive and functional. And you can see that we can both reline coronary arteries. This is CMFDA staining, so only live cells, large diameter and smaller diameter vessels, veins here, and even the trabeculae and endocardium. You can see the endocardium here. So again, large and small diameter vessels throughout this construct. And when we looked at thrombogenicity based on thrombomodulin activity, you can see that in fact when we reline our construct, we can get blood to flow through the construct to a much greater degree and inhibit clot formation. Now, what turned out to be even more interesting is when we transplanted this construct that we had re-endothelialized, you can see we recruit a large number of cells in here. And initially, although there's some clotting in the ventricle, we don't see a large amount of blood in the wall of the matrix, but we do see very large numbers of cells. And we thought that, in fact, perhaps we had an inflammatory response, but less than 5% of these cells are CD45 positive, CD11B positive, or WT1 positive. And the truth is the majority of the cells that migrate in seem to either be FLIC1 positive or PDGFR2 positive cells. And you can see here that they appear to be homing to a region like a vessel and surrounding the vessel here. So we're excited about the fact that our matrix can actually recruit cells when we heterotopically transplant it. So the next question we ask is, could re-endothelialization actually impact the function of these constructs? So we did a re-endothelialization in vitro for 7 days and then injected cardiocytes into the parenchyma of the construct. And you can see here that by day 10, after re-implantation of cells, we can now get construct that can generate force and actually has, in this case we used rat ventricular cardiocytes, and you can see that we get good pacing and we actually get an appropriate force frequency response. And then you can see in comparison when we use cardiocytes alone or cardiocytes plus endothelial cells, although these are early numbers, we actually see pretty significant improvement in function with the endothelial cells. Moving forward then, what I do want to point out is that pacing is required to get synchrony. If we don't pace these constructs and train the cells, the matrix itself is not enough to teach them to behave as synchronous cardiocytes. So although we think 3D is important, what we've found in our hands at least is if we can electrically stimulate these cells, and we will get them to be synchronous. However, physiology also matters. If we don't place mechanical load on the cells, if we don't put preload, afterload on this heart construct, we never get maturation of the cells. We only see microscopic beating. So physiology matters in this construct as well. I'll just show you that it's possible to do the same thing with human ES-derived endothelial and smooth muscle cells in endothelial cells in green, smooth muscle cells in red, and that you can see when we put these cells back in the construct that they coalesce and begin to form fibers that contain both endothelial and smooth muscle cells. Now, heart is interesting and fun, but the reality is this can be applied to any type organ system, heart, liver, lung, kidney, all shown here from rat. That in fact we can decellularize whole organ systems, the heart, lung here, diaphragm. You can see we can decellularize the whole gut here, the liver, the abdomen, you see stomach beginning to be engorged here. You see kidney here, the adrenal gland right there. And then in fact we can decellularize a whole rat. And when we do, every tissue is absent cells, eyes, brain, skin, muscle, even ovary, and even the tongue. And if you look at the tongue here, you can see that despite the decellularization, we retain even the taste buds on the surface. So I think we have a pretty robust three-dimensional architecture, and we can begin to think about building human organs. So to do that, we've said it's going to take a while to get here to end-stage matrix plus cells. So let's look at some of the benefits of matrix alone or matrix plus cells as a test bed for stem cell biology. So the first thing I want to talk about briefly is our ability to take this matrix and either make ring constructs or patch constructs that we can seed with cells and begin to look at physiology. And you can see here this speeds up. We can see drug responsiveness in these constructs, and that we can get cells to mature on these constructs. I can't tell if this is moving or not, but we actually, by 4 days after implantation on one of these rings, see microstructures. Doesn't appear to be moving. I'll try that again. Let's see. And then by day 8, we can actually see macroscopic contraction of the ring. And you can see here we can get cells to be pretty robustly and synchronously just in our ring structure. And we can now test drug effects and pharmacology and look at the effects of pacing or any other input that we want to examine in these rings. We can do this with rings seeded both with cardiocytes as well as with human embryonic derived stem cells and human iPS cells. What I can tell you is that in our hands, ES cells always behave better, mature earlier than iPS cells, but we're moving forward with this technology because I think it's going to be the most physiologically relevant. But not all matrices are created equally. When we take the same cells and we transplant them on either gelatin healthy matrix in sort of red here and infarcted matrix in green, you can see that in fact infarcted matrix upregulates a number of stem cell genes, whereas healthy matrix has a much more robust effect on cardiac-specific genes. So we think, in fact, we now have a test bed as well where we can begin to look at the role of matrix and whether or not, one of the questions is why cell therapy is not more beneficial and what effects we can actually begin to manipulate in vivo, and we think rather than having to go in vivo, we can do these experiments in the laboratory and begin to look at whether or not we can ever get full maturation on our infarcted matrix. In addition, we can ask what I call the architecture versus geometry or versus composition question. So we take the same intact three-dimensional matrix and we place cells on it and look at the expression of specific cardiac genes, and these are human cardiac progenitor cells, and look at a gelatin made from the same matrix, and you can see that the 3D matrix, as I would have anticipated, is much stronger at driving cardiac cell, in this case in KX2.5 and troponin T gene expression, than the gelatin. So 3D is better than 2D, 2D is better than gelatin and the normal matrices. We can also ask what I call the nature versus nurture question. We take cardiac-derived progenitor cells and we place them on heart, liver, or kidney matrix, and you can see that morphology differs here among the three organs. And when we look at gene expression, again, stem cell gene expression, myocardial early heart gene expression, or endoderm gene expression, that in fact myocardium seems to drive these cells to a greater degree than the other matrices, and the cells seem to know that they came from heart rather than responding robustly to the other matrices. We can do the same thing with pig matrix, and then the question becomes, can we do anything important in vivo? Our first approach to moving in vivo is taking decellularized patches to provide structural and vascular support post-infarction. So we took an infarcted rat on day 3, transplanted a patch, and this is a lousy picture, but it's an early picture showing that in fact, rather than getting this thinned out region of myocardium, when we transplant the patch, we see a much thicker region here. When we look at function, relative change in EF, we can prevent the decrement in function. Again, these are preliminary data. We've now got an N of 6 in each group. And at the same time, what turned out to be most fascinating to me is that this patch, which was previously completely acellular, is now vascularized and full of cells. And when we look at the heart, you can see that these cells are actually creating a channel moving from the myocardium into this patch. And much like you saw with the trachea earlier, we get vascularization. Now, I thought perhaps, again, we had an inflammatory response, but this is CD45 gene expression here, and you see very few CD45 positive cells. This is CD103 expression, and actually the next slide should have showed PDGFR2, which is by far the most abundant gene expression. So I wanted to show you the robustness of the process. The same thing can happen for lung, and you can see here rat lung and pig lung. We're working with Angela Mortari at the University of Minnesota to move forward with lung recellularization as well, and with liver. And I just want to show you here our early recellularization of liver and how quickly and easily it can happen with hepatocytes. This is real-time infusion of these cells. And we can actually keep the cells alive for up to a month. We can use rat or human hepatocytes. We've looked at midazolam and testosterone metabolism over time. We look at albumin and urea secretion and glucose. And you can see by histology as well that although this is cadaveric liver and you see the sinusoids and the organization, although this isn't perfect, we're beginning to get organization here. And at the ultrastructural level, looks very similar to cadaveric liver. Other people are doing it too. It's scalable to human organs. And I want to just point out here that not just liver, but kidney and also pancreas. This is duodenum and pancreas, so soft and more and muscularized tissues can be decellularized and retain their full structure. Based on that, I went to Spain a year and a half ago, and we actually began this process on human hearts. And you can see here the first human heart that we decellularized. So this is a human heart, and you can see the muscularized region here, the great vessels. This shows it better here, the muscularized region, muscle here, and fat on the surface of the heart. And then at day 3, much like our pig constructs, although we still have what looks like fat on the surface of the heart, there's no muscle left here. So you can see this is white, just like the pig heart, no muscle left in the vessels. This is endoscopy of that human heart, showing that in fact the interior of the heart is not muscularized any longer, that the chordae are intact. You see some brown there that originally we thought might be cells after 3 days. The pulmonary valve is patent. The right ventricle is acellular. What we found since then by pathology is that these brown spots are in fact lipofuscin deposits. You can see the valves are patent here, pulmonary and aortic valve. We did compression of media with our decellularized construct, and you can see the valves still work here. We can look at, by ultrasound, we can look at the wall of the myocardium and show that the fiber orientation that rotates through the wall of the heart so that the heart can squeeze is still intact. So you see these lines rotate through the wall of the heart. When we put a camera down the left anterior descending coronary artery, you can see the surface of this is intact, even though there's no, and you can see branching of a vessel here in the great vein, but we haven't damaged the surface of this vessel, which is shown better here when we actually show infusion of blood back into this construct. And we've done microbubble studies and echo studies to show we can do this at the small vessel level as well. To show convincingly that we've removed all of the cells, here's fat, normal fat on the surface of the heart. You see cell bodies. But I also want to point out the underlying lighter structures, which are the architecture of the fat that we typically ignore, that when we decellularize is what's left entirely. And that we don't see any cells left in the aortic valve cusps or the left auricle. We do see some detergent that we haven't fully washed out. This is aorta, normal aorta on the left with cell bodies, our decellularized aorta on the right. Again, intraventricular septum is completely acellular. There is some detergent left in the vessels that we're working on. And when we look at the ultrastructure, it's completely, you can see in the control we have cells lining these, pulmonary vein and aorta. In the decell, the cells are completely missing. So the real question was, what could we do with it? And I'll just close with this slide saying that we've taken a stepwise approach, and much like we did in the US, we said, let's see how we can use these cells to drive stem cell maturation. So we took some human mesenchymal derived stem cells, and this is human myocardium, and the matrix is actually fairly beautiful, I think, but I fully didn't expect what we saw next. When we put human mesenchymal cells back on this matrix, what we saw was a remarkable organization of the cells and a differentiation in a way that looks very muscle fiber-like. And I don't really know how to explain this. I wish I did. It's reproducible. We've done it multiple times. I didn't believe it at first, but it's true. And now we've got to go back and try to figure out how this works. So where are we? Cell therapy continues. The current focus is on new diseases. New cells are being discovered. Decellularization of complex organs is feasible. We preserve structure. The door is open now, I think. And within 5 years, I think we'll see part of a human heart in a first-in-human study. As you heard so elegantly earlier, trachea, which is a simple tube, has already been transplanted. Bladder, a simple balloon, likewise, and I would argue that going forward, the rate-limiting step in this field will be cells. We tried to overcome this within embryonic stem cells, and you can see here in one of our rat hearts where we transplanted embryonic stem cells, we were able to get a robust proliferation of these cells. Unfortunately, it's not good news. It's actually bad news because we don't see good differentiation of these cells. So I think we can say that we're going to do stepwise rebuilding, use the matrix alone, use matrix in vivo. We can also accept that it's a hard problem and go drink a beer. There's a huge amount to do, and I would just say that we have to be really careful doing this going forward and not move forward too quickly because this technology has a lot of promise and we really want it to recognize that promise. And I have to thank the people who did all the work. Thank you very much.